All right. Can people hear me okay? Oh, good. I'm getting all kinds of weird error messages from Zoom for some reason. All right. So where are we at? I think we finished up just here. Last time. Sorry. I had a puncture on the way into work, so spent an age fixing that on the side of the road. So I'm not my usual calm and collected self. I'm also bloody boiling as well. All right, so at the end of last class, we finished up talking about the end replication problem, right? And that's essentially created by eukaryotic chromosomes being linear, right? So if you remember, there are a few differences between, why don't I ask you? What are the differences between prokaryotic and eukaryotic DNA replication? So the central principles are the same, but what are some of the differences? Things that you find in eukaryotes that you don't find in prokaryotes. You know, one of them staring at you in the face. I actually just told you. What I thought about asking you instead. So I'll type that out while you're thinking. Different polymerases? Good. How? In what sense? That's a good answer. What are the different polymerases? Like, kind of, what are the difference? Oh yeah, Greek letters for sure. But aside from what they're called, how does what they do differ? I was actually just reading this before I started the Zoom, so I wanted to find something out. So let's start with the start of DNA replication in prokaryotes and eukaryotes. After the replication bubble has been opened up by helicase and single-strand binding proteins and the like, what has to be synthesized first? First of all, before any DNA replication can, or DNA synthesis can occur, you've got a bunch of them on the picture in front of you. Well, yeah, this occurs at the origin of replication. You're right, Isaiah. But once the DNA has been opened up, what has to be synthesized first? Give you a clue, it's not DNA. And it's not protein either, before anybody says that. Nucleotides, yeah, but what type of nucleotides? What are they made into? So it's a type of nucleic acid, and it's not DNA. RNA, right? RNA what? What are they called? Kind of old-fashioned, but small kids read them when they're learning to read. Find them in the casings of bullets. What else? You got one on the side of those little annoying yard engines, primer, like that. Yeah, primer. Thank you, Karen. So an RNA primer. So what synthesizes the RNA primer in prokaryotes? It's really easy to remember because it's kind of actually the word that Caitlin just typed up, with a few letters changed. What denotes an enzyme in biology? What three letters added to the end of something? Yeah, primase, right? So what synthesizes the RNA primer in eukaryotes? technically it is also primase, but I'll get into that in a second. Yeah, DNA polymerase alpha, right? So that's one of the key differences. And actually, I just found out this like, 9 minutes ago. DNA pol alpha, there's your Greek letters too, Stephanie, consists of 4 subunits. Remember, I always talk about these things as protein complexes, right? They're not single proteins. They're different proteins that have been added together, essentially, to make one big enzyme. And anyway, it consists of 4 subunits, 2 polymerase subunits and 2 RNA or primase. So though technically DNA polymerase has a primase in it, as a whole, it is DNA polymerase alpha that synthesizes the primer. That's one of the differences between eukaryotes and prokaryotes. In prokaryotes, there's a separate enzyme entirely called primase. Okay, so how much DNA synthesis does DNA pol alpha do? Oh, that's better. Does it synthesize the whole DNA, like DNA pol 3 in prokaryotes? No. Good answer. Why? What does? What does synthesize the bulk of the DNA in eukaryotes? Actually, there's 2 DNA polymerases, delta and epsilon. Awesome. And wouldn't worry too much about which is which, but why are there 2, essentially, Stephanie, or anybody else? Why are there 2 DNA polymerases in for the bulk of the DNA for eukaryotes? Right, prokaryotes only have 1, DNA pol 3. Good stuff. Yes, exactly. Leading and lagging, and it's actually the delta that does the lagging strand. But again, those kinds of details you can worry about in, other courses. What I really want you to get is the fundamental difference between prokaryotes and eukaryotes. Prokaryotes use a separate enzyme to make the primers, and then 1 DNA polymerase to make all of the DNA, right? Whether it's leading or lagging. Eukaryotes have 1 DNA polymerase, which just does a tiny amount. Literally, it's like primer plus about 20 base pairs. And then it hands over to either delta or epsilon, depending on which strand it's on. Okay, so that's a pretty fundamental difference. What else? What are the other differences? Sadly, that is finished. Yeah, that's good. Kind of treat this as like a study session in a sense. We've got a homologous recombination to do, but that'll take, I don't know, like 15 minutes or so. Want to make sure that you've got all this stuff down, lots of details. What else? There's like right in front of you on the PowerPoint in front of you, if you have that up. What other differences are there? Oh, I think my coffee's finished. Keep thinking, typing, pardon me, and I'll get my coffee. Now that I've cooled myself down a little bit with a can of pop. These things are things that I will quiz you mercilessly on. As in, without mercy. All right, what shape do prokaryotic genomes take? They're 1 chromosome. What's the shape of a bacterial chromosome? Yeah, circular. Actually, I was just thinking my kids are really into Squid Game on Netflix, which I'm not sure is entirely appropriate for my kids. But anyway, that was one of the shapes that they had to cut out of this like hard sugar candy. Anyway, so they're circular. So it just goes around in a circle. What shape are eukaryotic chromosomes? It's right there in front of you. I'll give you a da, da, da. Yes, exactly. Thank you. They are linear, a straight line, right? So what problem does that cause? Well, first off, they're different in size, right? So what's another big difference between bacteria and eukaryotes? Think about the size and how long DNA synthesis takes. Do prokaryotes have bigger genomes than eukaryotes? They're smaller, right? So what do you see in front of you on the screen? In addition to the fact that these are linear chromosomes, how many origins of replication do you see? 3. How many origins of replication do you have in a bacterial circular chromosome? 1. All right. So that's a consequence of eukaryotic genomes being far, far larger. Each eukaryotic chromosome is typically larger than a bacterial chromosome by at least 5-fold. So that's another big difference. What problem, which is again in front of you right here, does having linear chromosomes cause? You could even just copy and paste this into the chat box. What problem does creates, actually, yeah, that's a good point. That's another big difference. So DNA in eukaryotes are complexed with proteins, histones. Can anybody remember what that complex is called? DNA plus protein or DNA plus histones? Begins with a C. Ends with an in. Chromatin. Yes, exactly. Chromatin. All right, so that's one point. That's a good point, Stephanie. What problem do the ends create in eukaryotes? That's a good cup of coffee. Gaps. So those are created by the removal of what, Melody and Bianca? You're correct. Almost. Getting there, Caitlin. Not quite yet, though. So what has to be removed? Yes, primers. Oh, that's another difference too. All right, let's follow this one along, and then I've got to go back to primers. Okay, so you're talking about telomeres, Caitlin. What's the purpose of telomeres or telomeres, however you pronounce it? What do they exist for? Because they are largely repetitive junk, as far as I'm aware. So why have them when they don't do anything in terms of protein production? What's the purpose of telomeres or telomeres? Type it up too. Yeah, I want a good answer though, Katie. Not just a good question. I can do that. Yeah, so it's to prevent DNA from or the chromosomes from shortening, right? as I said, as a result from replication, because every time you replicate the DNA, one end, like the 5' end, will have a primer that has to be removed, right? And so when you remove that primer, you leave this gap, right, which cannot be filled. So telomeres act as a buffer, right, to prevent that shortening from eating into something important, like a gene. Okay, so how do stem cells get around that problem? What enzyme do they produce? Tumor cells too. Oh, actually, that's a good question, Kaylin. The primer gap isn't prevented by telomeres. It occurs in the telomeres. And basically, it doesn't matter because there are no genes in the telomeres. How do you pronounce it in the US? Is it telomeres or telomeres? Telo, I guess. T as in cup of tea. Come on, educate me. Poor lonely Brit in the US. Texas, actually, which is kind of another country too. Anybody? Oh, doesn't matter. So essentially, Caitlin, those telomeres are a buffer. They can be chewed up and shortened by DNA replication without causing any harm to the chromosome. Now, as I am sadly approaching middle age, are my chromosomes longer, or are my telomeres rather, longer or shorter than my children's? So if they shorten every round of DNA replication, exactly, they will be shorter. So as you age, your telomeres get shorter. So, oh, can anybody remember what the cell division limit is called in terms of numbers? I think it's about 60 cell divisions. Can we remember what that's called? It's a something limit. Begins with H and ends with ick. It's a Hayflick limit. So basically, those telomeres can't be allowed to get any shorter, right, after about 60 cell divisions. And that results in a really cool process, well, not cool from our perspective, but from a cell biology perspective, called senescence. Okay, so how do cells like stem cells and tumor cells escape that limit? What enzyme do they produce? Remember how to turn a thing that's produced into the enzyme that produces it? You add an ASE at the end. Yay! It's like DNA polymerase makes DNA polymers. RNA polymers are made by RNA polymerase. It's telomerase or telomerase. I don't know how you pronounce it. But anyway, that lengthens the telomeres. %100% correct, Brianna. Okay, so back to primers. Another difference between prokaryotes and eukaryotes. How do these primers get removed in either one? What's the process? And what does it? What does the job? All right, let's be a bit more specific. What does it in prokaryotes? Let's get back to the differences in DNA polymerases between eukaryotes and prokaryotes. What removes the RNA, sorry, and fills the gap with DNA in prokaryotes? Go on. Give you a hint. It's a DNA polymerase. Yes, good job, Isaiah. It's DNA pol 1. So what two different functions does DNA pol 1 have? It's obviously a DNA polymerase. What's the other? What do you have to remove before adding DNA? Wherever there used to be a primer, what do you have to remove first? So the first part, Isaiah, is not correct. So DNA pol 1 is not involved in DNA repair. That's 2, 4, and 5 from memory. But it does remove the primers. So the other function is an RNase. Right, as before, RNases degrade RNA. Okay, so how is this done in eukaryotes? Is it done the same way? By a specialized DNA polymerase? Just this bit left, and then we'll get on to homologous recombination. So what's the mechanism here? How does it differ? Yes, so DNA polymerase, and it's going to be DNA polymerase delta, right? And then ligase seals the gaps, but DNA ligase and its function is the same in prokaryotes and eukaryotes. What removes the primer? So Caitlin, you're correct. The DNA polymerase delta replaces the DNA that was primer, but how is the primer removed? Nope. Primase synthesizes primers in prokaryotes. It's one of the last slides, I think, one or two before this one on the end replication problem. Which one is it? Great, there you go. It's actually the slide before this. Okay, so just to recap, because the silence is a little deafening. That's okay, Caitlin. That's why we revise and study, right? So in eukaryotes, the primer is actually flipped out by a protein called replication protein A. That's this bit here. And then once it's flipped out, it gets cut off. That frees up this space for DNA pol delta to continue along to the end, which is what you see down here. And then that primer and DNA thing is cut up by exonucleases. All right, these are the kinds of details that you need to know for the exam. Right? Because the reason why I'm telling you these details is because they're really important to how this stuff works. And how this stuff works is really important to a whole bunch of different things. Right? So it's not designed to torture you, obviously, but it essentially gives you the foundation for a lot of other stuff. All right, so last thing. Okay. Can you hear me okay still? Oh no, I left my pens on. You numpty. That's like the second black pen that I've done that to. I have to get a whole bunch more now. My secret stash. You see my demon? Okay. So again, this is between non-sister chromatids. So we have, essentially, we have this, right? I'm not going to do this for all of it. That might help visualize things. Right, so essentially in prophase 1, it looks like that, right? We have two sister chromatids that have been essentially from one chromosome that's been replicated. Two sister chromatids here, homologous chromosomes, right? These two different ones. So the chromatids on the outside never undergo recombination. That is why you'll always get at least 50% parental chromosomes in any kind of cross involving genes on the same chromosome, right? Always at least 50%. And whether you get More than 50% depends on whether or not the two genes you're looking at are linked, but we're not going to worry about that right now. I'm not going to keep putting up the outside ones just because it takes a lot of space and it's kind of redundant. So there's my other pen. Oh, I hope I haven't duffed in my, no, I haven't. Awesome. So these are very precisely there, aligned by something called the synaptonemal complex, right? That's one of the, well, the main thing that actually creates such a precise exchange of material. Then the first thing that occurs, and it could happen in either one of these non-sister chromatids, we have a double-stranded break, also called a DSB. And this model is one of at least two, if not more, models on how this works, right? And so we don't actually really know exactly how this works, right? It's still a work in progress. So I can give you the theory behind, or the stuff behind one of those models, but it may not be exactly that in reality. It's kind of hard to tell until we do know what it is. Okay, so first you cause double-stranded break. Now, double-stranded breaks are typically catastrophic, right? So one way in which you can accumulate double-stranded breaks is to go swimming around in a pool of spent nuclear fuel rods, or live near Hanford in Washington State. They've got loads of them there, right? Or be a salmon in the Columbia River. But either way, usually when you get double-stranded breaks, that's catastrophic, right? And it affects replicating cells more than non-dividing cells, which is why radiation poisoning typically affects things like the gut, skin, hair, stuff like that, right? Those are the ones which, the tissues or organs which tend to fail first. So this is not catastrophic. This is required for the process, right? And so after that, and again, I'm just going to write up the ones here. So you have this double-stranded break here, right? Ah, there it is actually. Oh, let me tell you what, let me get rid of this so I'm bigger. That's better. You can see what I'm doing now. Now, once this break occurs, now we kind of chew back, maybe not that much, some of the DNA. So now we have these overhangs, right? Essentially single-stranded DNA, where before we had double-stranded DNA. That all make sense so far? Pardon me. Now what happens is kind of weird, to be honest. Essentially, this free end invades this chromosome here. What's hard to see, Kaylin? Is it me waving this around, or is it the pens being a little bit faint? Is that because of the reflection? Let me turn the lights off. That may help. Darkness. Like Batman, out of the Lego Batman film. Is that better? Oh, okay. Is anybody else having that problem? Maybe that you need to kind of expand zoom as well. Sometimes it crops when you have it minimized. Okay, cool. But thanks for the heads up, though. It's always good to know. So now the inside, or the strands on the outside, remember, we're still talking about the inside non-sister chromatids. Like so. Now this strand, oops, this should be broken here. I'm sorry. Invades the other strand and base pairs. It's complementary, right? Oh, pointed to the wrong thing. And in doing so, it forces out the original strand. It kind of creates that kind of bubble, essentially. This is the strand invasion part. All right, let me take a picture of that and then I'll rub it off and continue. I actually learned just yesterday how to sync my phone to my computer, which I was super impressed myself with that. My wife can actually believe that I managed it all on my own, which is kind of funny. All right, let's get rid of this. Okie dokie. So now that invading strand gets synthesized using the non-sister chromatid as a template. And now, I left this pen uncapped last night, which is why it's looking a bit flaky. Now we have this kind of displaced strand, it's probably the best word for it, is now able to base pair with the other strand on the invading piece of DNA. Right, so remember this one had a free end kind of poked up there and base paired with the non-sister chromatid, it's complementary strand of that, and goes along, displaces the original complementary strand until it can base pair with the other strand of the non-sister chromatid that was broken. Now this acts as template. There's the black pen. Until you get to the point where it's gone pretty much as far as it can. And the other strand gets, oops. What am I doing up? There we go. Synthesized like so, right? Until basically the strand being synthesized hits the end of the broken piece, right? And the same up here as well. These then get stuck together. Like so. with DNA ligase. And now we have structures that are called holiday junctions. And they look just like that. So it actually looks like a cross-shaped, right, where those two chromosomes are held together. Now, depending on where that cut occurs, right, you can either cut it, let's use a different color. You can either cut there and there, or there and there, whole bunch of different combinations. And when you do, you end up with a range of different possibilities in terms of DNA. And those are cut by holiday junction resolving enzymes. There you go. That's pretty much it. So again, not particularly well understood. It's not a super easy concept to get either, to be honest. But the basic process, as far as we understand it, at least for this model, is one strand gets cut, degraded back a little bit, and that free end invades the non-sister chromatid, and that is then used as a template to synthesize a bit of matching DNA, essentially, which then gets cut. And that is homologous recombination. That's how it actually works. Let's take a picture before I forget. Oh, yeah, yeah. So I'm still here, just in darkness. Essentially, the exam will be same format as last one, so exam 1. 40 questions, you're going to have an hour and 15 minutes, which is not a long time, to take at any point during the day on Friday. I'll be in Zoom. You don't have to attend, there's no lecture, but I'll be in Zoom from 9 to at least 9:30, maybe even 10 o'clock. It just kind of depends when I remember to check if anybody's there, just in case you need any help understanding the exam questions or last minute check of what you know, that kind of thing, right? We'll be there. And yeah, study, practice all of this stuff, right? Try writing these processes out yourself, right? Without using your notes. Like write out or draw even better, draw out the flow chart, I guess, of DNA replication, right? Eukaryotes or prokaryotes. Any of that kind of stuff will help you figure out what you don't know. Obviously, going back through your notes and the PowerPoints and these videos will be helpful, but it's still not going to get you exactly where you need to go, because that's kind of passive learning, right? Let's just listen to me chunter on and make bad jokes, right? What you really need to find out is what you don't know. Because once you know what you don't know, you can fix that problem, right? You can go and look it up or ask me questions or whatever, right? but you have to know that you don't know it first. So focus on that, practice your 3-point crosses, right? They take time, and there will be a bunch of questions on that stuff. And yeah, that's pretty much it from me for this section. Does anybody else have any questions? All right, everybody. Well, good luck. See you on the other side. Being fast.
Sir, I had a question
Oh.
real quick.
Go for it.
Can you hear me?
I can.
So I know today we have lab, I think, but I wasn't really feeling well, and I missed the previous session before, so I'm not sure what I should do.
You know what? Just hang on for a little bit, and we'll talk about that. I'm just gonna stop the recording.